Hey, everybody. Hey, Peter. Sorry for the delay here. Just waiting on Manu still, but figured I would just get us all started. Might as well just start hanging out. Hello, Akil. How are you? Oh, see a message here.
Yeah, hi, Tyler. Hi.
Good to see you. I'm just going to send a link to Manu real quick.
Hey everyone, very happy to be here.
Hey, Zabir.
Hey Taylor, how are you?
I'm good. I'm good. How have you been?
I've been okay. How about you? You are well? Are you well?
Yes, doing well.
Great, super nice.
Apologies for missing the. Hey, how's it going, Pranav? Yeah, apologies for missing the past sessions. I know there's been some craziness going on, but
No
looks
problem.
like we're good to go. So yeah, very excited to see you all here. Hello, Emma. All right. I guess while we wait for Manu. Any interesting discoveries this week? Anything frugal or fun to share?
One thing I can add, it's not related to what we have been discussing, but it's frugal. So we want to measure rain because the monsoons are here. So how to measure it effectively? So the frugal idea is to use this clicker, right? And add a Pythagorean cup to it, which is a cup that drains itself once it is filled, right? So every time it drains, we know that exact amount of rainfall. And so how many times it has drained in a month would give us the rainfall. So just
That's very
thing
clever.
that we do. Yeah, let's see if it works.
Very interesting. How much force do you need to put on the button? Do you have to do
Yeah, so
any?
that has to be adjusted. That has to be adjusted. So we'll add some weights to the Pythagorean cup to make it just to increase the precision of the device,
Yeah.
right? Because we want very fine measurements. So uh, the other idea was that you use some electronics, but then once you add more complications to it, it doesn't spread well amongst the people. So we want to make it very simple.
Yeah.
I apologize for a rather vulgar question, but is that how the toilet flusher mechanism works in a way?
Exactly. It's a
Yeah,
Pythagorean cup. Yeah, the
super cool.
bigger version.
That's awesome. Super. Anyone else? Any other fun things y'all have been thinking about? Doesn't have, yeah, just it could be anything interesting.
Oh, I've had a random question. So, where were we supposed to upload our homeworks? Because I have a
Oh,
few, and I've done some experiments with paper planes that I've documented on Notion. I just
let's go.
send
Okay,
them.
this is fantastic. So if you want on the class Notion, there's a place where you can link your Notion page.
Mhm.
Under the, what's it called? It's called the team roster, I think it is.
Yeah, that's where it is. Yeah.
Okay, so you've put yours there.
Mhm.
Okay, fantastic. So I think that should be the place. And I think the plan, I'll check with Manu, but I think the plan is basically we're just gonna, I mean, we've been taking a look periodically, but I think we can just take a look at the Notion links that are here. And I think it'd be really fun to do a share out not only of the project teams, but also of the homeworks, because I know a lot of you guys have done some pretty cool stuff in those that are on the side. So
Yeah.
I'll check with Manu, but so far I think that is the right place for it to be.
Sounds good.
All right, Divyanshi. Oops. Oh yes, I see questions from Subir. How are the paper planes, Peter? Did they fly and how far?
Yeah, so the goal was to make something that moves on its own, right? And I figured since we can have a stream of air, and we were talking about those where they were caterpillars that have a propeller and then they move. It was on one of the Fridays, I believe. I think Anton was talking about it a lot, right? So I started looking into things that move on their own that I can just build easily within like 5 minutes. And I realized I can build a bunch of paper planes. So the first thing I've built was a paraglider with origami, and that thing didn't fly out of my hands like completely. It would just tank right away. So I started like building more and more intricate paper planes, and I was looking up YouTube tutorials and stuff like that. And then I realized they're not exactly moving on their own, right? I'm actually throwing them. So I pulled out like a room fan, and I decided to start launching the 4 planes that I've built and the paraglider. I decided to launch them out of it. And what's interesting, the paraglider, given the airstream from the fan, like the paper plane that I will like launch out of my hand, the one that flew the farthest, right? It would never actually even pick up on the stream of air from the fan. But the paraglider that would just tank if I throw it, if I would just kind of like let go of it right above the fan, it would actually fly a few feet. So that was a revelation.
That's very interesting. That's very interesting. I
Yeah.
wonder, yeah, I guess it's like something to do with the front air, airstream or the aerodynamics from the front angle of attack versus back angle of attack from your fan, huh? Or what do you think?
I actually think it has more to do with, well, we can't exactly say weight because all of these things were actually built out of a single sheet of paper, so all of them would have the same weight.
Mm,
But the distribution of the weight and like how big of a wingspan they have actually plays a fairly large role, because like a paper plane that you want to shoot out of your hand and you want to have it like fly the farthest, it actually has to have like a pretty good attack angle, and it has to have fairly small wings. It just has to sustain itself in the air. It's not trying to like actually use the airstream to move. But if you're trying to like have it use the airstream to move, it has to have fairly large wings. And also, I want to describe it as density, but it's not exactly density, but just basically has to do with the amount of paper matter in a certain volume of air, if you know what I mean.
maybe like a mass distribution kind of question of where the mass of the plane is located.
Yeah, so the more distributed the mass over like, over a bigger, not volume. Yeah, I think it has to do with the wingspan. The bigger the area of wings, the better it performs when there's like something that pushes it as far as the air goes, right? And smaller the wings, the better it just like holds up in still air.
That's awesome. I love this exploration. I was recently just looking up more on this topic. I remember Manu referencing the theoretical experiment of flying these paper planes out of the International Space Station and seeing if any could be recovered from the ground. And it was funny because I feel like the reason, at least the stated reason that I was able to find that they canceled the project was because they couldn't figure out how they could track the planes properly to find out if any had survived, which seems like such a trivial way for the experiment to fail. Yeah, more work for the space debris team, exactly.
I mean, uh, have they thought of attaching like small GPS modules to them? I mean, they probably wouldn't be able to track them very well until they enter the atmosphere, but when they enter the atmosphere, like they don't fly fast enough to actually burn.
Yeah, although actually these ones would, I think, because they're entering from vacuum. So I think they'd be coming in at like Mach 7.
Hmm.
But the paper should be able to, well, they have some fancy paper that they at least they made, I think, that theoretically should survive.
Yeah, honestly, I just don't know physics well enough, but like I'm not even sure. So if you launch a paper plane out of the space station, I mean, in theory, since it's in the vacuum, there should be no gas friction with it. So in theory, it just should infinitely fly unless it like reaches uh some gravity or an atmosphere of some kind.
Yeah.
Like,
Yeah, I think their hypothesis or their plan was to do something very silly where they would like take it from the International Space Station and then, or approximately the same altitude, and then delay, and then slow the orbit. So basically, you're not going to be actually in this perfectly circular orbit. Hello, Manu.
hey, Manu.
We're
Hey
talking about flying
everyone.
paper airplanes from the space station, but now we can get on started with projects.
Yeah.
Yeah, I mean, that is an actual experiment. I don't know if you guys have read that paper. It's quite a puzzling. Yeah, I think this is from the equivalent of the Japanese equivalent of NASA, where they demonstrated that you can actually have a paper airplane enter the orbit because of kind of the velocities won't be that large. And they actually simulated. The only problem they had was if they launched and the paper plane landed on Earth, how will they ever find it? So first of all, so good to see you guys, and also some of the old faces as well. I'm sorry I've been out a little bit. My dad got sick and kind of I had at this exact same time I was juggling calls with doctors in India, which is never fun. But I think he's good now, and it's yeah. I think I know I had to move this 2 times, 3 times, and that must not be fun for you guys. But apologies from my end. So we could turn this into, I think maybe one thing that would be first useful would be is to go through projects that people are thinking about. I'm assuming that was one of the agenda items, but then we can also have an open-ended discussion, and maybe we can go through a round of people who are ready to present something, and then take that into an open-ended brainstorming session. Whichever way people want to run it, I'm happy either way. I now have basically a couple of hours, so I'm happy to stay late tonight.
Yeah, that sounds great. I wonder, yeah, I think totally up to everyone. Maybe we can just have an open floor if anyone has anything interesting they want to be discussing immediately. And if not, then we can go straight into presentations if that works. Because I do know, I think folks had planned to do them last week. So I expect folks, I'm guessing there's going to be a good amount prepared if you guys feel like doing that first. Should we just do a show of hands, like in favor of doing presentations first? Hands, presentations. Okay, I'm seeing a good amount of hands. Should we do that, Manu?
Yeah, I think that's a good idea.
All right, let's do it.
Oh, Divyanshi, go ahead.
Oh, I think that may have been a hand.
Oh, got it.
Okay, fantastic. Who wants to volunteer as the first team? And you should be able to go ahead, Pranav. Awesome. Yeah, go for it.
Yeah, we can start.
Yeah.
Okay.
It's awesome to see, Pranav, that you were awake in the morning and also are awake now.
Yeah, I know. I'm also in the PST time zone, so
I see.
it's not
Okay.
it's not super
Not
late.
too bad. Okay,
It's
that's good.
not too bad. Yeah. Okay. Let me share my screen. Can everyone see my screen? Okay.
Yeah.
It's loading. But okay. Maybe I'll jump right in. So yeah, our team is called erythrocyte because we're trying to image red blood cells, which are erythrocytes, for detecting sickle cell disease. And we hope to use a low-cost automated microscope for this. And this is one of our concepts, but I'll get to that. Our team involves, Divyanshi, Kiran, Mohammed, Rishika, Sana, Thoma, and myself, Pranav. A lot of us are here today. So yeah, they'll join for discussion, and some will join for the presentation. So in general, sickle cell disease is an inherited blood disorder which affects red blood cells or erythrocytes and causes them to be form these rigid sickle-shaped cells, which can block blood vessels, cause pain crisis, and even death. It has been estimated that In the next 30 years, around 15 million babies will be born with sickle cell disease. And unfortunately, a very high percentage of those children, so 50 to 90%, could die before the age of 5 in low-income countries. This sadly is due to the lack of diagnosis and treatment options in these areas. This map shows the number of newborns born with sickle cell anemia. Sickle cell anemia is the severe form of the disease. So this is due to the inheritance of two genes, so two mutated genes from both parents. The asymptomatic case is actually called sickle cell trait, which is due to the mutation or the inheritance of one mutated gene. And this map actually is incomplete due to two reasons. One, it doesn't show the sickle cell trait cases, and it also doesn't have the complete picture for cases that lack screening. So a lot of the low-income countries lack screening, so the complete picture of the prevalence of the disease is missing. For our project, we're focusing on rural and remote communities, and right now we're taking examples of Nepal and India. Some locations are shown where the prevalence of the disease is high. Just for some rough numbers, the rural population of Nepal is in the order of 24 million, and in India it's much higher, 900 million. The prevalence of the disease in these rural areas is not fully known. There have been some studies that study or find the prevalence in some of these communities, and in Nepal it has been found to be up to 25% in some communities, whereas in India, in some of the rural communities, it's up to 40%, so it's quite high. As an example, these are what health posts look like in Nepal. So this is in one of the visits. These are two health posts which do the screening for the sickle cell disease. You can see microscopes, some reagents, and some of them are run down, unfortunately. And this is where the screening is done in places like these. And the microscope is used for a test called a sickling test, which I'll talk about a little bit more in detail. And the gold standard test, which is used to diagnose sickle cell disease, is actually more than 150 kilometers away, at least from these health posts, and it could take more than 3 hours. So it's, in terms of accessibility, it's not very accessible for the communities. Comparing the health posts, so the rural health post versus a clinic or a hospital which has the gold standard test, which is an HPLC machine, we know that the lives of people with access to either of these are quite different. So anyone who actually tests positive in a screening test requires a confirmatory test from an HPLC machine. So they would have to have their samples either sent to the hospital or themselves travel to the hospital. In Nepal, at least, the government has provision for around 1 lakh rupees for treatment of people diagnosed with sickle cell anemia, which is a positive step. But there are, because of this diagnostic gap, not a lot of people are screened or have been diagnosed with sickle cell anemia. And there is this gap of screening for people with sickle cell disease. And again, the situation is very different if we look into aspects of geographical, in the geographical context. So access to the HPLC machines or good screening facilities, financial situations in rural communities are usually worse than in urban areas. and other social considerations. Some of it being resistance to screening in the first place. So if you go to a rural community and say, I want to draw 3 milliliters of blood from you or your children, many people are hesitant. So children or even healthy adults are not usually willing to give blood for screening. There's also added social stigma associated with the disease due to lack of awareness. So people are not sure whether it's a genetic disease or an infectious disease, and I mean that it's those are two different classes altogether. And we've seen that awareness of the disease actually increases participation. So this year is like an awareness campaign that was done by UBC Sickle Cell Project a few years ago, which increased the participation in this like mass screening test. And in terms of frugality, it should cost close to what is, at least for the screening, what is close to the current screening techniques, which is around less than $1 per test. And this includes the cost of the microscope lumped into, let's say, screening a whole community, for instance. And ideally, this should be supported by some government entity or an NGO, because as I said earlier, people are not willing in the first place to give blood for screening, especially if they are healthy. And I want to stress that it is important to even screen for people that are healthy, because they might have sickle cell trait and pass on their mutated genes to their children, causing sickle cell disease. So other considerations, so accessibility, it has to be all the screening and all the awareness campaigns have to be done in the local language. For example, in this community, which is in Nepal, they don't speak Nepali, but their own language, which is Tharu. So it has to be in their own language. The first original problem statement that we had was, can a low-cost microscope automate the scanning and detection of sickle cell disease from images of red blood cells during a sickling test? Now, what is a sickling test? It's a simple test where you add a drop of blood into on a slide, mix it with a reagent, which removes the oxygen and causes sickling in people with sickle cell disease, and doesn't, or people that are healthy have their. Sorry, is anyone seeing something? Oh no, okay. Yeah, people that have are healthy, they have their cells unchanged. Yeah, we had this table as well. I won't go into much detail, but we had tried to divide the function into, let's say, automating the scanning and focusing, automating disease detection, and possibly smartphone microscopy. Now, this is again our original table. We've added other options, which I'll get into. We can get back to this table if we have time. But some frugal prototypes and objectives for this course. To start with, we can try observing sickle cells using a low-cost microscope such as Foldscope. So this could be our, let's say, first prototype. Then try some automation for either scanning, focusing, using platforms like Squid or Octopi. We could have community conversations, explore other parameters like viscosity, and even other parameters related to general flow of healthy blood versus sickle blood. So this could be in a microfluidic device, a low-cost microfluidic device. We'll have Sana join us for this first prototype.
Yeah, okay. So hello everyone. So as mentioned by Pranav, we have categorized our prototypes and our objective. So the first prototype, which is an realistic approach, and if we have a full scope, we can, by using Lancet, we can just prick a blood and we can see inside the foldscope. So basically, it's something, if you have a foldscope, we can differentiate between anemic people RBC count and healthy people RBC count. But the main purpose of this is we should have some agglutination agent, because whenever we do a agglutination, test, we see in the microscope that is using 10x and 45x. But using foldscope, like I have a foldscope, I know how to prick a blood using Lancet. I can take people who, the samples from anemic patient, and I can use my blood, and I can compare how the agglutination takes place. Is there any difference between a healthy and an anemic people RBC count, and what's the agglutination pattern? And this is the realistic approach that can be frugally done within this. I think till first week of June we are planning to do this, and the main focus is the agglutination pattern. And this was inspired by Professor Manu Microsome's blog. Pranav, the next slide. So basically, I just found out the credit was Manu Ji, and what he observed was RBC observation with iodine, and this was the third trial. So inspired by this work, can we have this using a foldscope, and is it possible to screen out and to see the agglutination pattern, the difference between agglutination pattern, and is it possible to do it? Of course, if you are using a Lancet, we need someone who's expert or who has done internship in pathological thing. So this is the first realistic approach we are thinking, and if someone has foldscope, we can go for it and we can upload on the Microsome's blog. And it is really exciting to work, to physically, practically work on this. Yeah, so it will be continued by Pranav.
Okay, thank you, Sana. So yeah, the other aspect was the automated microscopy. So yeah, the squid and octopi platforms, these are actually a very good fit for a solution like this. For this course itself, I mean, there could be some potential of testing some components, so imaging, motorized scanning, autofocus, some of these. In terms of implementation, some automated system like this can be implemented before field trials for, let's say, automating scanning for faster data collection, which could be for machine learning purposes. And during field implementation could be for automating scanning and disease detection to reduce the need for highly trained personnel. Other options could be for exploring or looking into community conversations, interviews in India, because we have a lot of team members from India, identifying the need, current practices, any idea about the awareness, prevalence of the disease, the level of frugality required, potential challenges, and so on. We also had other alternatives in mind, so microfluidic calculations. So one of our team members had this idea of having a simple microfluidic device that has a coded and an uncoded channel. So a coded channel that causes sickling and an uncoded one that doesn't. And then we can use that to differentiate normal sickle cell trait and sickle cell disease. And then, yeah, I mean, just trying to run some calculations to see if something like that is feasible. Looking into viscosity, agglutination, like Sana mentioned. And I mean, yeah, this is one that I could think of. So even doing educational materials for sickle cell disease awareness, I mean, this could be done in different languages and so on. But yeah, this is all we have so far. We're looking forward to your feedback. Yeah, I'll just stop sharing my screen for now.
So that was fantastic. I'm assuming, do we have some time? How many other groups are presenting today? Because I think based on that, we can figure out how much time. Do we have another group presenting after this? Peter is presenting, and then Akhil and Emma, or are you guys all in the same team? Two
Me and
teams.
Emma are.
Okay.
Yeah, me and Peter.
Okay, and then Akhil. So two more. Maybe let's do oh, Divyanshi, were you going to say something? Just a quick set of questions / ideas. So first of all, I think what's very important is to get an intuition for the role of throughput in identifying. And this was at the heart of why for malaria we automated, because it's a rare parasite and we need to be able to really actually see around. For one parasite per microliter, we did the calculation. We needed to image somewhere of the order of 1 million to 5 million cells to make sure that no matter what, we will always see a parasite in a single slide. But sickle cell is the completely opposite. Goes back to the Fermi. How many fields of view, how many cells do you need to see? And if it is true that that phenotype is visible in every single cell, then we have a very straightforward approach in which we will move towards resolution and high magnification imaging, but with no scanning capabilities, because every drop, every few sets of cells are deterministic. So I think this is one thing, and my intuition in this context is that is true, that it is deterministic, especially when you're using a sickling agent, which is not the case traditionally if you don't use the sickling agent. There's also a new objective that we are about to release. And I think if you look at the original Foldscope paper, we have described that objective already. I have a few, so I'll get a few of you to everybody in this team to test and also just give you guys some data, because from a resolution point of view, we can go all the way down to 0.7, which is phenomenal for imaging single blood cells because they are quite large. And so from a detection point of view, that would actually be straightforward. And then the question that becomes is building and thinking about the kind of slide that rules out any concentration differences, how the sickling agents are allowed, and then repeatability. So that would be one approach. I think as in experiments, I should make sure that I get you guys some of these materials right away. The second bit around this would be access to real samples. So at this moment, Pranav, do you or anybody in the team, do you guys actually have access to sickle cell bank?
Yeah. So I do. Yeah. As a disclaimer, I mean this is part of a larger study that we're planning. I am planning for my PhD, and right now I do have access to a hospital that provides blood with sickle cell disease, and then I have been doing the sickling test.
And then is that at UBC?
Yes, I mean it's affiliated with UBC.
Yeah. And I think, does anybody else in the team also in parallel has access to sickle cell blood? Just so in case, as multiple experiments are happening in two sides of the world. Not yet, right?
Yeah, I do have access, but yeah, it's the hospital, it's nearby. So I can.
Yeah, but I think, remember, there are challenges around patient samples and things. So it has to be done in an IRB context. And sometimes there are de-identified sample banks that are collected in which IRB doesn't have to be filed and everything. So when I said patient samples, I meant through all formal means, not just getting access to the sample. But on the other hand, there are banks. So having a conversation with a set of doctors is useful, because I think when you guys are all thinking about a breadth of ideas, because several sets of ideas have been in the past looked at in terms of how blood spreads in paper. This is one common example that's given. But what you want to do is really push for, if you're going to go down the direction of screening, you want to make sure that you choose what is the level of sensitivity and specificity that you want to assign to it. And with screening, and especially when the kind of treatments might be fairly complex, for example, or the outcomes can be complex for a patient, it's very difficult. I mean, the common example that I often give is, for example, mammograms. Although on paper, it appears that, we should just massively screen. it's actually quite complex. The benefits come about if the technology for detection is really good. But if it's not so good, false positives completely overwhelm the kind of a system. So doing a little bit of a calculation on identifying what is a good enough target. And then lastly, I think what was kind of daunting to me when I look at these numbers, that 40% prevalence and that 20% prevalence, and then focusing primarily on, say, pregnant women or certain cases in which if that number is so large, you end up thinking a little bit about choose a population framework in which there are urgent action items, and that'll determine whether the setting that you are working on is completely rural or semi-rural or also urban, just in the early phase of the project, just so that you can imagine. I mean, I think it's very straightforward to thinking about directly applying Octopi to this because we have opened the platform. We are now at the scale of manufacturing this. So I think one thing that we should do is, Pranav, if this is going to become a core portion of what you do, is to get you one of those platforms. At this point, that's straightforward.
We
But-
did talk to Hongshan actually, and then he mentioned that it would be, I guess, like mid-June is a realistic time frame.
Yeah, I mean, it's good, but I think that all also just comes from now, since you know, as these things rise in priority, there are certain sets of things that, you know, because we make them ourselves, we can make them, and you know, not only we, you can make them as well, and so it's all doable. We should do that sooner rather than later. This should all be driven by when is the first time someone in your team is actually going to a clinical setting. And let's work backward from there to figure out when is the first time. And then ideally, initially, you should not shut down, but actually test on all the platforms, because you might realize that in a certain setting, you would need throughput. And in another setting, you would actually need no electricity, absolute frugality. And so multiple solutions already would actually be very valuable. So let's work backwards on this to figure out what date might be a good time to be in the field. And then the last thing, which again goes back to if you can identify the number of cells you need to be able to see. Another framework to think about certain things like these is also driven by, if you think of what we do in Planktoscope, we never make a slide and we have cells pass by a detector. Now, sometimes you might think that, oh, you need to actually resolve a cell to know its shape. But if you're just scattering light across a cell, just these deformed shapes would scatter light dramatically differently. And so then it's kind of a single particle detector. It's not even an imaging system, just a tiny laser and a photodetector, and thinking a little bit about whether anything has been done in that space, which is a common principle of how flow cytometers work, but at a macro scale. That might be another fun orthogonal idea, primarily because it provides both throughput and frugality if there is no or very little optics. And you might imagine that you want to be able to do this with a large number of cells. And I'm just questioning whether imaging is actually even necessary. So that would be another fun thing. I'm wondering whether have you guys read anything in that space? Whether when people take sickle cell blood and you pass it through a flow cytometer, for example, what does the backscattering signal looks like? Yeah, I mean, this is the idea of a laser projection microscope. I don't like them for imaging because too much information is lost, but this is exactly what is so beautiful about blood. And only when you image so much blood, you sort of start realizing this, is blood is incredibly uniform. Like basically the remarkable commonality of the size tolerance of your red blood cells is so precise. So when you start seeing these other I'll show you guys one image, which is kind of fun in that context. And then I think I'll pass that. Okay, while I search for this image, let's other people, questions, comments. I think I'm just excited about this. So let me pull out this image while, so I think there are a couple of to-do items just from this discussion alone, but I'll wait for.
I had one comment regarding the number of cells. At least in my tests, what I've found is that the sickling at least is not uniform. And then, I mean, some cells sickle. I mean, there are some areas that sickle more, and then it could be due to either like heterogeneity in the cells itself. So it depends on the age and stuff. It could be also in the sample preparation. So some areas might have more of the reagent, and so at least within a slide, we do need to look at different fields of view just because of the heterogeneity in the slide.
Yeah, and then the question then becomes, is it a heterogeneity in the cells, in which case I agree with you, or is it in the heterogeneity of how you apply the reagent, in which case there might be another simpler solution, in which just adding a drop might not be the right way to actually introduce the sample. Okay, other question. I'm going to go offline for just 2 seconds. I have to just take care of something, and then I'll show you guys that slides. But you guys, you guys should just go through any other questions that people have for the.
The floor is open. Also, Pranav, I guess just a general one is sort of like what are there any like big existential questions about the approach that you're like, you know, on the team that you guys are currently juggling, or something that I think it could be fun if there's anything big just to have everyone think about and discuss?
I guess we are having I mean, there are different approaches that we're considering. So like even like committing to one or like committing to all, like that's the one thing that I wasn't sure about. Like, should we just like divide and conquer, or should we all like try to focus on one problem? Or I mean, it from my perspective, it does make sense to maybe divide and conquer, but I don't know.
Yeah, I think a comment on this is early enough that don't rule out, you have to explore. So you're in that exploration phase. You know, the fact that the quarter ends has nothing to do with evaluating sets of ideas. So it's not about, oh, we're about to come to an end. This is really the beginning of a project per se. You have to have enough data to convince yourself that a certain approach is not sufficient. So I would be in the camp of you should, especially because many of the imaging solutions that you might actually test and try validating for this have already been built. I mean, it took us 7 years to get to where we are now and where Octopi is, and around 10-ish years to where Foldscope is. And the moment you see the next version of a Foldscope, you'll immediately realize the images look very different from what you have even seen before. And so don't rule out anything. I would argue that expand, but make sure that you have a very concrete action plan on those sets of experiments. And especially because people are distributed, having multiple people choose certain approaches would actually be very valuable. Here is a picture that I want to show you guys. So this is not published data, but this comes out from the newer version of Octopi. So just as a background, you've all seen the 2019 version. Now there is the 2021 version.
A quick question: what is an octopi? If you could just give like a
the object that you're looking at right in front of your screen is an octopi. It's a modular microscope that we have been building towards for malaria, TB, lots of diagnostic applications. It does automated scanning of glass slides, for example. I think currently you can think of it as an intermediate between microscopy and flow cytometry. So as a platform, this image is something like 5 million cells in 2 minutes or something. So essentially think of, and it does fluorescence, bright field, and phase. And the point of modularity is that there are different modules. So there will be basically a sickle cell module, which is a minimum system, or this is a TB module, or there is a malaria module. But the thing that I wanted to show you guys from a context of data is now if it ends up being a situation where we need a very large number of cells. So what you're looking at are these two data sets where we have registered back to single cells that have parasites. And of course, this is data focused on parasites, but this is going through, this is a movie that will continue for many minutes, and it's even identifying specific shaped cells, for example. And so what your input point is in these thin smears are the shape of these cells, and they're all rounded. So throughput is, it's very doable. It just really, you have to ask the question backwards. How many cells do you need to see? And in malaria, we know for our sensitivity and specificity to be 1 parasite per microliter, we need to image around 5 million cells. And currently, humans that do this type of a test only image between 3 to 10,000 cells. So you can see what a big difference automation actually makes. And then one last point, and then let's jump to the other side of the story, is to give you an example of how uniform and amazing blood is. So this is an entire scan of a slide, and it's, I don't have the gigapixel image of this, but this entire thing is, this is 25 million cells on a single slide. And in the gigapan video, which I'll send you guys a link of, you can zoom in into every single image. And I don't think you need that type of a number of cells to identify. But when you look at this image, the most striking part about this is how uniform individual cells are. It's, you know, I think PowerPoint doesn't allow you to zoom beyond a certain point, but this as an image actually has around 25 million or so cells. Okay, I think yeah, it's unfortunate. The power cell point pixelates the picture so you can't see it. But I'll send you guys this raw data. Just so if you want to look at cells, even from a single human, and see, oh, what is the baseline variation? That would actually also be very valuable. Yeah, let's, other comments, let's switch to make sure that we have everybody else covered as well, and then we can come back to this as well. Yeah, I think, Subhir, a couple of people have tried the attaching imager to a 3D printer's head. One of the, I mean, it's good for macroscopic imaging. One of the problems is really around the repeatability and the focus side of the story. What blood cells do require is, you know, a blurry image versus being perfectly in focus. And so the mechanical stability while being stationary is not something what 3D printers will. So there are better solutions, better adapted solutions, while the 3D printers are fantastic for macro scale imaging. So maybe around, if your goal was 10 micron or so resolution, there's some really fantastic ways of imaging worms, imaging flies, imaging ants. They are fantastic ways of doing large scale macroscopy. And I think, yeah, I would say for blood, there are a couple other options and stages which can still utilize 3D printing. And I think, yeah, this is slightly different from thinking a little bit about the the print heads have gotten really good now, but there is this context of both flatness index. If you have to refocus every single time, or can you just go about moving place to place without refocusing all the time? Okay, so let's switch. Maybe Peter and Emma, do you guys want to go next, or Akhil?
Oh, sounds good. We can go next.
Yeah.
All right. So we are Team Frugal Biolaboratory, Frugal Biolab for short. That's probably better. Let's see. Let me make the Zoom window as little as possible so it doesn't disturb anyone. But yeah, this is the people that are doing this project with us. I am Peter. We have Emma, Apeksha, Divyanshi, Kiran, Akifa, Mohammed, and Sana. They're with us. And the question that we're trying to answer is, how can bioenthusiasts or underfunded scientists establish a lab at home or in the field using commonly available household items? And how can we help them overcome the social and structural barriers in their journey to run a lab and contributing to science? So partially we might be a couple of years late for for us to have an enormously huge impact with our project, because COVID, well, we've been saying that COVID is coming to an end for a little while now, but we're actually experiencing another wave. So yeah, but there are no more lockdowns probably happening anymore at this point. But one of the things that happened during the lockdowns, and I've talked to some people about this, and it turned out that some biology PhD students, they actually had to run their experiments at home. So that could be another target audience for this. And one of the problems that we've actually discussed last Monday in the group was a situation when someone was trying to use, they found out that you can sterilize things, essentially replacing autoclave with a pressure cooker or a yogurt maker. However, they didn't understand the physics of air and water vapor under pressure before they went into it, and they've pressurized their whatever it was, I don't remember, let's just say it was media, but they pressurized there, it was media, and it still came out with contamination. Some of the things of how to repurpose household items into a laboratory, while some of this knowledge is out there, there is not too many resources available on how to specifically do this, so it's reproducible, and it can be done by anyone pretty much anywhere. So what we're trying to go, what we essentially are trying to do, we're trying to propose a way to do these things in this environment. And if you take a look at the picture on the right, the setup is incredibly frugal. Like what you see over here is just like some 1 liter jars. And I mean, if you just like ignore the glassware over here, if you look at the things that are on the wall, what you see is just some light bulbs and tape and aluminum foil and maybe some saran wrap and pins and just basic paper towels along with some reagents. But well, there's probably almost no way to get the reagents the super frugal way, but this is a topic for a discussion, right? So basic laboratory setup is rather fairly cheap to do a lot of the basic laboratory procedure. You can build an autoclave out of a pressure cooker with almost no modifications required. You can use polypropylene microwave oven safe food containers instead of petri dishes, because petri dishes are actually made out of polypropylene too, and they're heat resistant, so this is amazing. And these food containers are also dirt cheap. And actually this one on this particular image is 12 millimeters, 120 millimeters in diameter at the top, and 100 millimeters in diameter at the bottom, which kind of is basically the size of a typical 100 millimeter petri dish. Just keep on repeating millimeters. This is a standard petri dish, like the most easy, like the most common one you can find. And actually prepare for preparing the media, you also don't really have to go and use a lot of fancy lab chemicals that you can only order from a supplier. You can literally just go buy some agar powder, which is very commonly available all over the world. Which yeah, and it works amazing. For your proteins, you can get some cornstarch, and for your carbohydrates, you can always use sugar, because what is dextrose? It's sugar. You open if you take a bottle of lab dextrose and you look inside, you're literally going to see sugar grains. Like this is what it is. And with things like foldscope, you can even do microscopy just using your smartphone, which is very, very frugal. And if you're looking to do more advanced experiments, and this is where we can actually probably collaborate with other teams, and we already are starting like a little collaboration with people from Friendzymes. Yeah, there are more advanced things that you can buy fairly cheaply. You can build a Frugal Pi, which is a fluorescent imaging station, for about $200. Very, very frugal, an amazing piece of tech. You can build a bioreactor from plastic bottle, which guys from Friendzymes can tell us about. You can get a DNA sequencer, which is kind of pricey, but it's still a sub $1,000 device. You can PCR machines for under for about $600 are available on the market, and you can even repurpose a piece of a broken computer mouse into a low resolution. And by low resolution, I mean 8x8 pixels, but a very high frame per second camera, so you can capture even like the slightest movements. Yeah, so a bit about what it's going to cost for the end user. It should be $500 or less to put together a basic biolaboratory. And we understand that $500, for example, in the United States or Western Europe, is not the same as $500, say, in Uganda or China or India, or really like this amount of money is pretty arbitrary. The point is, it just should be affordable. It should not eat into a underfunded scientist grant very much, and it also shouldn't. And also, a citizen scientist should be able to afford it. We should probably avoid spending more than $1,500 to achieve most single tasks. So, for example, if the goal of your Frugal Biolab setup, for example, is to sequence for example, is to sequence molds and figure out like what particular contaminations you're dealing with here or there. I don't know, it's just an arbitrary task. It should cost you less than $1,500 to get everything set up and running. And if we take a look at some numbers, we have 8.8 million scientists worldwide, and those are like legal, not citizen scientists, but like people who are actually like publishing and working in academia. And about 80% of the research comes out of the G20 countries, and which leaves potential for 1 million scientists to be underfunded, because not all governments are so easygoing on funding science. Yep. So, and I will pass this off to Emma, and she's going to explain the importance of this project.
Yeah, so as Peter was saying, like limited budgets, and then how that varies across the global scale. So trying to make it more cheaper options so it's more accessible to everyone, but more accessibility directly to technology isn't just going to magically make every single science experiment be aimed towards repairing a wrong in the world. From there was a great study I read recently that the more access to ancestry tests has actually sort of enabled white nationalists to solidify their ideas of racial purity. So it's really important that when we're pushing for more accessibility of these implements, we're also pushing for more ethical use. of these instruments. Next slide. And then this is the functional. Actually, could you go back one?
Yes.
Oh no, go back to the other one. Sorry. Yeah.
Mhm.
Yeah, so the functional, not spend too much time on this. So we aim to provide an ability, sort of an actual protocol to help people actually design a singular experiment in the field and the materials that will be needed, because you can do a lot of work with you know, just a petri dish and some agar and a microscope. That can be one single experiment. So just helping people to actually be able to understand what's needed for particular tasks. And then to do that, we're going to create guidelines to help that happen. And then another quite important thing is identifying the structural and social barriers surrounding the biotechnology. So I aim to have a qualitative list of questions prepared to send out to biohackers, anyone that's interested, bioenthusiasts, basically anyone that's interested, and to see the social barriers that have stopped them. See if you just go to the next slide, Peter. Go to the next one. They're all in a different order. Yeah. So how we identify the social and structural issues are really important to how we center like justice back into our practice as scientists. And to do that, we're going to investigate the social and structural barriers, ensuring that people are educated about the ethics. So hopefully from the qualitative research, we'll see what exact barriers could be. Emotional barriers. The field of academia is very hard to penetrate if you're someone that isn't used to the language that's used. It's very inaccessible, and that can create a lot of emotional barriers for people that actually to gain the confidence to start thinking that they can conduct their own experiments. And it's really important to see what they are, so we can create guidelines to help people through that. And then see if you just want to go back to the global challenges. I'll talk about that, and then I'll give it back to you. Or do you want to go through this, Peter?
Or actually, yeah, I would love to go.
Yeah, you go through this.
So since we're creating this for a global environment, so we are actually trying to we're trying to do this so it's accessible to pretty much everyone in the world. So of course we would run into some challenges. So one of the challenges, the three main ones are, the first one would be consider the cost and the accessibility of components in different parts of the world, right? Like I've mentioned before, like a value of $100 in the US, it does not equal to a value of $100 in Thailand. In Thailand, for example, you can get a lot more stuff like say like a lot more groceries for $100 than in the US. However, you can probably buy less stuff that is imported in Thailand rather than you can buy rather than in the US for with the same $100. And as an example, we have a Raspberry Pi in Australia that actually costs 52 American dollars, right? And in the US it only costs 35. I believe you just have to pay the tax on top of that, but the shipping is free. Like I personally went through a process of buying a Raspberry Pi recently. But yeah, in Australia it's $52, and like sometimes you have to pay the shipping fee. Yeah. Protocols should be number two is our protocols should be adjustable to geography and weather. There are very different climates around the world, and even like the geography of the US would be very, very different. But to keep things a bit simpler, and just to give you an example, there are places like Cyprus where the climate is extremely humid, right? And there are places like say I don't know, Las Vegas, where the climate is extremely dry, and evaporation would be much lower in a humid climate. So we can just tell someone to let something evaporate for 20-30 minutes because evaporation depends on the humidity. So we have to take geography and weather into account, like other challenges that can arise over here. As far as geographical and weather ish features go, is altitude and different pressure, air pressure, temperature, and so on. And probably the most important one, at least to me, our protocol should be gear agnostic. So for example, quart jars, which are, a quart is 946 milliliters for everyone outside of the US, which is probably useless information for you anyway, but quart jars don't exist outside of the US, right? But liter jars are in abundance. So liter jars should perform as good in a protocol as a quart jar would. Yeah, so it doesn't really, so it shouldn't really matter whether you have a metric or an imperial system, or what particular style or geometry of jars your country uses. It should be geared, like, yeah, the things that we talk about in our protocols should be agnostic to local variations to the components. Yeah. Implementation. So the way we are planning to implement it is we're going to take GitHub and we're going to use it as a storage for the protocols themselves in a markdown format, and for to display those protocols and to actually make this thing accessible to everyone worldwide. We are building a website. Part of it is already live, but I can't share the link because it's just filled with dummy data right now. It wouldn't make sense, but just to show it off over here. And the reason why we chose GitHub is that it's a global platform for people to collaborate on things and contribute, so their name is actually saved somewhere, and then there's a pretty good history of who made which edits. And we want this thing to be open source. I come from open source software, and I'm a big fan, and I would love this to be a global standard. So people would be able to easily contribute their protocols, just the global community outside of us through GitHub. We will be able to moderate it and add it to our collection, and all of the stuff is going to be licensed by Creative Commons by license. So it's absolutely free to use in commercial, non-commercial projects, whatever. Do anything with you want, do anything whatever you want with it. Just give us some credit. That's all we ask. So yeah, our goal is to create a platform where that's going to be easily accessible to read for people. And it's also going to be fairly easy to contribute to if one would believe that they have something to contribute. So these are our next steps. So one thing we should do is meet, and we've already kind of started working on this, meet with scientists that work in the field or with lower funding to better understand their needs, and also scientists that had to work in kitchens during COVID. Very interesting statement, but nevertheless. So, and the questions that we want to ask them is, what do they wish they would have known before they got started, and what barriers do they still face, and how they have tackled the barriers that were in front of them before. And this is one of the things, Manu, that we actually want to ask you for help. If you know any of the scientists that work in the field and actually have to deal with extreme environments, it would be extremely valuable for us to interview them and ask them a few questions about how what research that they have to go through to establish to establish a workplace for themselves in their field environments. And if we can help future scientists that are also working in the field or are underfunded, so yeah, how can we help them with our resource? And the other step that we have is create a few fundamental protocols to fill up our website that would describe basic biological procedure. And this is literally in the span of this course, whatever time we have left. And to create the set of fundamental protocols, we need to figure out what procedure would be considered basic, right? And understand that there is obviously sterilization and maintaining sterile environment, because for a lot of science, sterile environment is pretty necessary. Media preparation and basic culturing, right? So that would be something to start with. And what else? How far should we push it? But yeah, that's our presentation. Thank you.
So quick set of reactions, questions. I mean, I think, yeah, the notion of testing the idea by interviewing a broader group of people is very straightforward. And already in the room here, you can all see that we all have different types of labs, or lab access for that matter. That's already a kind of a starting point to get a sense for that. Not all labs are, and sometimes I think it's valuable to first draw it out in what dimensions are labs different. So cost was one dimension, but there are also other dimensions. I mean, the second one is, of course, people that work in the field. It is not about cost, it's about portability, and it's about being able to bring your lab to a setting where biology is, sometimes the environments are extreme by definition. So I think the way that I would structure this, and again, it improves and increases the value of the proposition, is that sometimes it's not just about cost. It is also about being able to do new science. When you bring a capability into a new environment, that type of a question could not even be asked before, no matter how much. For example, I know of a scientist that regularly climbs tree canopies looking for new insects and new biology, and they go with a full scope, because no matter how much money, they can't climb with their Zeiss rappel hundreds of feet above. And I mean, it's just a new environment. And it's, so I think drawing that out would actually be very valuable, give you guys a sense of how you can connect what happens in the field to what you might want to do that's cost effective. I think one thing to do also just would be, as a comparison, is to just see lots of people have explored these notions of biolabs as hacker spaces and things. And I think most of them have a cost has not been a criteria as much. I've seen really fancy biolabs, for example. We've also seen one people labs. It's useful to create kind of a repository of what has been done, so that you can use what people have listed out, but also focus on what does not exist. And that might come from making also a list of protocols, that what is a minimal list of things that you want to do that this lab will enable? How do you draw that line? And that's a tough one, because science is so open. Like, if I really want to do viral infections in a cell line, suddenly there is a class of things that you have to worry about, that otherwise you have contaminants and other things. While if you even want to transform of a genome of an organism that's really hardy, you don't need that type of sterility. And so I think thinking a little bit about explicitly describing functionality and be driven by the function would be very valuable, because it'll also allow you to understand the limits of the idea itself. And when you find a limit, then you can overcome it. Like, I just said, sterility or temperature control in a lab. But maybe there are better ways of doing sterility than how we do it currently.
Mhm. Mhm. So essentially, it would be a good idea to collect what has been done in this field of yeah,
Yeah,
and
but also collect the kind of a minimal subset of protocols. Like when I think about, for example, Fab Labs, what they do is you can literally get an Excel sheet, you can download it from the Fab Labs website that has every single, down to the screw, every single part that goes in a minimal Fab Lab. And of course, what they have are called super Fab Labs that are
Mhm.
over the top and are even better than most engineering labs. But the basic minimum Fab Lab is an Excel sheet that you can download as if you're downloading a lab. So what would that kind of look like for this? And also mapping it to when you go into a machine shop, you have a good intuition for what you can do and what you cannot do. And so for a biolab, many a times that is not so explicit. And it's by design, because that field we're still discovering what you can and cannot do. It's not so obvious. Like some very clever biochemistry, and you can do incredible things which could not even be done with the fanciest of instruments. Like, sequencing was like that. It's just insane to think about that, where in places we can actually do sequencing. But choosing some targets in that space would actually be very valuable.
Mhm. Mhm.
Other comments, questions?
There's actually a full chat that we should I will actually look through right now, see if there's there's something that I can't answer right off the bat. Depth of the microscopes, okay, octopi,
I think you should go from the bottom up.
Okay, sounds good. Uh, okay, yes. Asana says create a frugal kitchen lab sterilization using a cooker. Got a lot of goof ups. Right. So there we go. There's a person who can benefit from these protocols right away. Kitchen scientists are a very interesting subspecies of a human population. Orange pie, I'm not sure it's software support. Raspberry Pi selling in black market for $200. That is true. There are.
It's just a Raspberry Pi. Raspberry Pi is not available.
Yeah, I've seen this problem before. There are a lot of like resellers of Raspberry Pis that do sell them at like an insane markup. The trick is to find like official like the official retailers. And over there, you would always find the basic model of the Raspberry Pi for $35, no matter what. Because I think they do have an agreement with the Raspberry Pi Foundation that they're not gonna mark it up more than that. And yeah, the Raspberry Pi market is actually a pretty ridiculous thing, and it probably deserves its own, not its own protocol, but it probably deserves a separate instruction on how to like source a Raspberry Pi for an adequate price. And same goes for many of those like small but super powerful computers. For example, Jetson Nanos. Those things are very, very unavailable. And I think Google TPUs are also in that like price space, which is very unfortunate. But yeah, people should be more aware of that, and I think we'll try to.
Pranav, go ahead.
Pranav has
Pranav, you have a question?
I had a question about your project. So I was wondering about like biosafety and biohazard and stuff like that, because I mean, to get access to a biolab here, I mean, in a university, you have to go through sets of training and I mean, and I mean, there's another aspect where if you do things in your own home or to your own self, then I mean, you take more risks, right? And uh, the I guess how do you address that aspect? Like, how do you make sure that firstly, I guess the users are safe, and then this being a bio lab, like you could potentially make biohazards not just for yourself but for your community, and even let's say bioweapons. I mean that's an other extreme, but
Yeah, so yeah, I see what I see what you're saying, Pranav. And if I go back to our wonderful table, if I can start the slideshow from the current slide, that would be amazing. Start from over here. Yeah, so we did look at the risk, and we assessed it as a low. Very short and simple. If handling with safety level 1 microorganisms, right? If you are not working with something that is potentially hazardous, if you're a citizen scientist, you probably should not work with something that is very hazardous right off the bat anyway. And the more you work, when you feel comfortable handling, I feel like if you're not comfortable doing certain things, your brain will just like naturally stop you from doing that. You would just not want to do it, right? If you, yeah. But generally, citizen scientists, in what I've seen, they don't really work with organisms with safety level over 1, right? And even so, source for citizen scientists or someone who is who doesn't have any sort of training, a person who doesn't have any sort of training would probably have a hard time sourcing a hazardous organism anyway. So I feel like the limitations have already been set in that regard before before even like the idea of this project. And I think that's actually enforced by governments one way or another. But nevertheless, you can still get your hands on some pretty dangerous stuff. Fortunately or unfortunately, I personally am all for it.
Yeah, that's what I aim to address with protocols around the ethics and sort of actually getting people to. I think it's something that's really lost in like any sort of scientific method, like the social sciences and even just history and anthropology, of how thought is actually brought is like completely overlooked. Like there's so many cases where science is just used to perpetuate colonial and capitalistic narratives, and that needs to be broken apart. And there's a fantastic non-binary biohacker called Mary Magic, and they argue they have an open source protocol for hormones made out of the pheromones of urine, and they argue that the safety of what they're doing is in direct comparison to the mass hormone hacking that has been that's been going on through the capitalistic system, through the input of like all these plastics that are causing like estrogen releases and stuff. So it's how do we create social protocols that get people to look at their scientific research in an intersectional way that doesn't perpetuate like harmful norms? And then when it comes to like the handling of sensitive things, it just needs to be like put into a protocol so it's accessible, so when people aren't getting themselves into trouble when they're actually doing something that's a safety level risk above what they might be capable of.
Yeah, and one thing I want to add is that there's definitely a risk of mishandling of the instruments, right? And there's always a risk no matter what you do. There's always a risk. And for example, if you're performing a very basic sterilization, I don't know, your pressure cooker might explode because the pressure inside of it is too high, right? But it also has like a special valve that prevents it from it. But let's just say that that might potentially happen. And there are all these risks, and the idea is to design the protocols in a very simplistic, have the protocols designed in a very simplistic manner so that there are as little dimensions to risk as possible and have all of those areas covered.
Yeah, and I mean, I think just one comment, and then let's switch to Akil and team
Mhm.
because we're gonna run out of time. Is
Sounds
there are
good.
also legal frameworks and laws that, so as citizens, law-abiding citizens in the country, there are some very specific laws around things, and science in the frontier pushes those laws, and then new laws are put in place. So what happened, for example, in the case of CRISPR in human germ lines led to lots of sets of discussions, and this is how society evolves as well. And in some sense, making what is possible accessible to people is also an important goal beyond the advances in science itself, because we have made an assumption that only a few group of people are capable of advancing for greater good in humanity. So, all of these risks have to be discussed in the light of what is to be gained as well, which is a society that is driven by science and logic. And I think some of you saw the most recent shootings. And if you go back and look at that manifesto, for example, there are citations on very directly on certain papers. So it's being discussed in the science community, whether just even regular scientists when we are publishing. So it's not just about somebody would generate evidence that then would be supported. It doesn't matter whatever scientific evidence is generated, even from traditional labs, that can actually be used and weaponized to spread hatred. And that is something that it doesn't mean that, science still has to be produced, but we also have to be ethical around. And there were lots of questions around whether those sets of papers did not clarify for the broader audience what it meant and what it did not mean. And what does it mean to actually publish papers in that space, which then could be weaponized? And when things are weaponized, how do we respond? This is a topic that goes far beyond what would happen in bio. I mean, I would separate, these are very important topics, but they have a small intersection, but they are bigger by themselves, just in general. And I think, yeah, I resonate with what Subir
Yeah.
said. It would be fun,
Well,
Emma, to
I
have
mean-
a broader discussion around this as well.
Yeah, with all honesty, almost everything can be weaponized at this point, one way or another. So, if these resources make their way into hands of more people, at least there's going to be more understanding of of how these things work. And even if they are weaponized, if people do have an understanding of how these things work, they might be able to de-weaponize it, if that's not a tautology, right?
Yeah.
But
And then
yeah, because
just for
knowledge is power.
clarification, I
Mhm.
was using that word weaponize much more as not as a literal sense, but just as morally
True.
weaponized. And but on the other hand, that literal sense is also true.
Mm.
Okay, on that note, let's move to the last talk, so we make sure that Akhil gets the time as well. So Akhil, do you want to jump in and share?
Yeah, sure. Yeah, so shall I start?
Yeah.
Yeah, so we are a saliva diagnostic team. So our team members include Janisa, Savitri, Sushma, and myself. So we started with a broad idea, that is development of a saliva-based kit for diagnosis and monitoring of systemic diseases. So we narrowed down to chronic kidney disease. So what is that disease? Like a chronic kidney disease, if we define it, it is a disease of kidney where we'll find abnormalities for a considerable period of time, like which is like considered more than 3 months. If we have an abnormality in kidney with a glomerular filtration rate of less than 60 ml per minute, and if we find any of the chronic kidney disease markers, then it is considered as chronic kidney disease. So what are these markers? So the markers, like we can broadly classify into like 5 or 6 subtypes, like one of which will be like a connective tissue abnormality, where you will find necrosis in most of the kidney tissue, and where the whole discoloration also will be seen. And there will be structural abnormality as well. And you can also see albuminuria, like albuminuria, where albumin content will be excess in urine. And then the glomerular filtration rate will be less, as I explained in the definition. And also the electrolyte concentrations, the sodium and the potassium electrolytes will be abnormal. So you will find cases such as hyperkalemia, hypernatremia, where potassium and sodium concentrations will be extremely high. And there will be sediments in urine as well. So these are the typical chronic kidney disease markers where you will find in any abnormality of kidney. So if we see the global preference of the disease, and if we compare it from like 1990 towards 2020, then we could get an idea of how the disease has like conquered the whole globe, where the incidence and prevalence, it has shot up to 87% and 89% when compared to like 1990. And if you see the death of in case of chronic kidney disease, it is like more than like 99%, where around 1.2 million people are getting affected. And the daily drivers, so the drivers of the disease, like it could be population growth, and it would also be due to aging, and as well as disorders such as diabetes. So if we see the global prevalence, then we could see that countries where there is a very high population rate, that is getting affected. Here we can see it's India, China, and Bangladesh. These countries are having a high incidence of this disease. And if we get an update of 2022, like we can see that like approximately 1 in 10 person is getting this disease, and it is more prevalent in women and elderly. And there are typical causes for this disease, including diabetes mellitus, hypertension as well, which is common as well. And if we see the death rate, we could get a clear idea why this disease is extremely important to address. We could, here this graphic shows that the rank in cause of disease, if we see from 2013 to 2040, the disease has been like progressing a lot, and we could see that it will be ranking 5th in the overall death estimate in the year 2040. That's what the estimate says here. And the large burden is mainly in the low and the middle income countries. So the underlying cause, if we see for a chronic kidney disease, it's like around like almost all the systemic pathologies are also involved. And in this case, we can see that diabetes type 1 and type 2, glomerular nephritis, and hypertension, these 4 contribute mostly to the occurrence of this disease. So these things, like we thought of having, we brainstormed the session, and then we came to a conclusion as to how to address this problem. So we went through the existing diagnosis and the monitoring strategies for chronic kidney disease. And if we see the strategies, it is a normal blood tests will be conducted, followed by normal urine tests, where they will typically check for the creatinine levels, then the blood urea nitrogen levels in both blood and urine. And then they will check for the electrolyte imbalance, that is the sodium and potassium levels, the homeostasis, whether there is a balance or not. And apart from that, they will also check for the filtration rate. And then apart from these two serological and the urine tests, we'll also go for a kidney ultrasound and kidney biopsies. So this is the normal routine diagnostic procedure for monitoring as well as diagnosing these disease, like this chronic kidney disease. So if we see the disadvantage of these tests, like in terms of long-term monitoring, the invasiveness of these blood tests will definitely come to play, as well as the cost factor for long-term monitoring is an issue as well. And it also requires trained professionals. So drawing a conclusion, like we could clearly see that there is a need for a low cost and a non-invasive method to diagnose and monitor diseases such as chronic kidney disease. And also, it would be great if we develop an easy to use at home kit for monitoring the progression of this disease. So we can also check for the prognosis of this disease. We can monitor the prognosis. So what solution can we get from this? Like, so the question is, like, can we develop a low cost device for diagnosis and monitoring of CKD using saliva as a non-invasive specimen instead of blood samples? So the reason why we took saliva is because like most of the biomolecules which are already found in blood and urine is also there in saliva. And also it mirrors the body's health. So this is the major reason why we took saliva as a non-invasive specimen. And here, like we could see several systemic biomarkers. And most importantly, it is the uric acid biomarker and the salivary electrolytes. And we also have creatinine, albumin, and other protein molecules as well. So we did a brainstorm session and came through this table, and I will go through it very quickly. First, we would like to address the saliva sampling methods. Like we would like to optimize the method of taking saliva, like how it can be optimized so that we can get a dynamic, like since saliva is dynamic in nature, we would like to have a standardized protocol to get an acceptable level of sample for testing. And then we came to the next step where we would like to work on the pH, viscosity, and the flow rate of the saliva, and just to see whether there is any variation in systemic diseases such as chronic kidney disease. And for that, the risks majorly involved this circadian variation in saliva. And the countermeasure would be to have a universally acceptable reference calibrator, and also to check to have a low cost device where we can detect the pH and variations and also the viscosity. And then we wanted to go through the salivary biomarker quantification, as well as the quantification of the electrolytes, how the electrolyte concentrations vary in diseases against the healthy individuals. And then we would like to also design and develop a low-cost, frugal device. Majorly, like we thought of implementing a paper chromatography-based assay or an ion complex formation assay to address this issue. And for that, we would have to optimize these isolation techniques to identify and address these biomolecules which are involved in this disease progression. And so the experiments which we are planning to perform, like during this course of this study, like we are planning to start with the detection of creatinine in saliva, because creatinine is the major biomarker in case of chronic kidney disease, and it could be easily detected by means of a simple complex formation with copper sulfate. And we would like to identify and calibrate the detection limit, because the detection limit of saliva is extremely low when compared to that of blood. So that calibration we would like to perform, and we would like to address the issue. And then we would like to validate this experiment initially in the lab in artificial saliva, and then we would like to take it to the next level on human saliva samples. And then we would like to develop a paper-based device, initially with like for creatinine detection, and later on we are planning to take it to the next step where we are planning to detect levels of electrolytes, sodium and potassium ions, as well as urea. And in making these detections, we could also estimate the saliva albumin creatinine ratio. So in the next 2 months, like we would like to address the first issue, that is the detection of creatinine in saliva, and then we would like to move on to the next half. So yeah, that's it. We're open for questions.
This was a fantastic piece of work. Yeah, I think you guys have really thought through about the challenge, and yeah, I think beautiful set of identification. So let's start with just on the literature side. Is there any work that has been done in the past for CKD on saliva? I mean, in terms of thinking about both biomarkers and just where does the literature stand? Is it in the discovery phase, or would you think that there are certain indications that saliva is a good place to start and it's really the implementation that's missing?
In case of saliva, like urea detection, they have done. Detection of urea from saliva, they have already been published actually. But in case of the electrolytes and creatinine, so far, no.
Mm-hmm. And then can you say a word or two about just everybody in the team or anybody else as well? What's the biological origin of creatinine? How does it make it into saliva as compared to other body fluids like sweat versus urine? Like, I mean, I'm just trying to anticipate a little bit. Of course, saliva is a fantastic, large volumes of sample is available, but what percentage or numbers, for example, when I was to think about a home test, when it really directly competes with urine? So would it be an order of magnitude lower in volumes as compared to urine? Like blood, I know blood, it escalates to a different level, but comparing urine and saliva.
Yeah, I think it will be very low. It is like 10 to 100 folds low actually.
In saliva?
Yeah.
Okay. So that's actually a good key insight to keep in mind around the ease of use that you provide by switching to saliva, while urine is also, it's a little bit more invasive, but not completely out. I think blood definitely meets the criteria that doing a blood test at home with those large volumes just is not possible. And maybe if it turns out that the set of tools and technologies, especially on the detection level side, gets harder, then there is a trade-off there. While I can also understand from an adoption perspective, there would be a group of community where saliva they will do, but urine nobody would. Just, human behavior. And if you're really trying to do this as a screening tool, spitting into something is fairly easy. Going to the bathroom, peeing, and then handling that in a normal home is not so easy. How do you dispose it? Just all kinds of things come in mind. And then just as a background, I mean, I think one thing that maybe when I do a one-to-one with this team, it's useful. For the last 2 years, I've been working on a saliva-based diagnostics, of course, much more for COVID. And we learned quite a lot, actually. Just initially in our IFUs, we write very specifically that people should not have coffee or orange juice or some things before. And when we ran the usability study, we had that written in the IFU, and then we asked the question whether you did it. And still, maybe I would say 5% of people checked the box that, yeah, they read it and they still did. So and we had to change our detection mechanism to be less sensitive to pH, because we could not rely 100% on the fact that people would actually follow this idea that don't have coffee and then immediately spit it out. I mean, the pH goes off the roof. The other thing we learned, and I think this is not applicable here, was just also blood and saliva. For some of the later stages of patients in COVID, there was enough blood in the sample that it made it difficult because it interfered with some of the reagents. But we had never thought that we would see blood in saliva. But again, I think just COVID was a tough case in that scenario, but I don't think that's applicable to you. Other questions, comments?
Hi, Akhil.
Go ahead, Kiran.
Yeah, so I would like to ask the same question with the line with Dr. Manu, that do we have any kind of proteomics or transcriptomics data which will suggest how CKD is affecting the levels in the saliva? I mean, somehow the map of saliva in context to diseased and undiseased condition. Do we have that in literature?
Actually, I'm not sure. I will go through the literature and I'll check it out. Yeah, it's a very interesting strategy to start with. Yeah, the proteomics
exactly.
data.
Yeah, and what I was thinking is, designing a tool and then thinking that what threshold or, the level of creatinine in the saliva, you're going to decide based on your device, which you're going to build, right?
Yeah.
But I think if we don't have the data, then there is another first step, in which you have to, thoroughly look into the level of creatinine in a certain population. I mean, there will be enough population number, and then go ahead with your device and then settle down how, again, the sensitivity and the specificity will look
The
like.
calibration part, right?
Yeah, exactly.
Sure.
Yeah, but I think maybe what Kiran is trying to say is there is the basic science, and then there is, no matter how expensive or how hard it is to detect or build a device for detecting it in saliva, the first thing is just a clinical study that unequivocally proves that the saliva mirrors what's happening in the disease. And that has nothing to do with the device. That has nothing, we can use $1 million tools to truly establish that there is a disease map to it.
Because sometimes what happens is, it depends upon the disease severity or, the percentage where the disease is falling, if it's beginning or it's in the severe form. So the changes are quite visible in the blood, but because the creatinine levels are very low in the saliva, we don't know how the changes in the bloods are going to get reflected in the saliva. It might be a possible scenario that when the condition is very, very severe, then it is getting reflected in the saliva and not in the beginning stages. So we have to even compare our saliva data with the blood data, that in this particular condition, the level of creatinine is different in the diseased and the non-diseased. In the blood is this, and in saliva is this. What is the difference you're getting? And that's how you need to, I think, proceed, because in my opinion, saliva is not going to reflect the disease condition that easily.
Yeah, I mean, sometimes there's only one way of finding out, is to actually, and then it's valuable to dig in to see whether any of those studies have been done. But on the other hand, I think this was a discussion we had internally in the last one-on-one meetings, or the last Friday discussion. If you look through that recording, I had made an analogy, which is a perfect example to give here as well. There are two types of approaches in tackling or kind of advancing the state of affairs. One is you focus on the hammer, and then eventually a different nail might come along And so in that hammer, in that context, is really the saliva is something that, I mean, it's absolutely wonderful. And whether it happens that CKD may or may not fail, that doesn't actually matter. There are so many targets that it can be then utilized in a broad range of applications. And that's a hammer approach. There is also the nail approach, which is being completely agnostic about how you tackle the problem, but the problem is what you're married to. And it's not one or the other. So there's no criticism around. Sometimes, as engineers and as scientists, it's perfectly okay to be passionate about a sample and then really saying, that's the hammer approach and the other one is the nail approach. What's problematic is if you stay with the hammer and the nail simultaneously, because then the trains don't meet. And I think these are sets of questions, are not a criticism, but are actually a way of just approaching this, because one of them, when you stick with one, that's perfectly okay. And pivoting on an application is also perfectly okay. But I think clinically, what we learned, which was very interesting, is we just wanted to get access to some saliva banks. And unlike Pranav, who can just go to a bank and get sickle cell blood samples, there are no banks for saliva. It's actually was quite surprising when we started this. And this is like 2 years ago. Maybe now it has changed, but it would be very valuable of, do we even have saliva of normal patients in a broad range of physiological conditions? When it's dry, my mouth kind of dries up. Maybe the proteomic contents change completely based on the weather of the day. But that's also true for urine. That's actually also true for blood. So it's not as if you can say that these types of things are completely agnostic. But it's valuable to think about where are we in the gold standards for databases. Maybe a few comments. Go ahead, Kiran. Did you have a comment?
Yeah, I have another question. Sorry.
Go for it.
So Akhil, you mentioned that you guys are going to monitor the sodium and potassium levels too, right? So do you think that the change in the sodium potassium levels are going to be directly the indicator of CKD or not the other factors?
It's not only CKD actually.
Okay.
It's like a change in the homeostasis, right? The imbalance.
Right.
Yeah, so that is one of the markers actually. That is not a specific marker for CKD. That is one of the markers for any systemic disease actually.
Okay, sister. So you're going to correlate that how every factor is there in the disease and control. Okay.
Yeah.
And another question that's very basic. Why did you go for, why did you choose to go with the saliva and not urine for this project? It's just.
Yeah. Actually, we thought of the approach of taking a sample, right? In some cases, getting a urine sample will be a little difficult compared to saliva. In case of long-term monitoring, in case of a deceased individual, taking saliva would be significantly simpler compared to urine. So that was one of the reasons why we went for saliva and not urine actually, because we are planning for long-term monitoring.
Kiran is challenging the same thing that I had also just challenged earlier, that I think with social context in mind, I actually agree with you, Akhil, that when you add the practicalities of although urine samples are also quite non-invasive, I think for several of the STD diagnostics projects in the class and a lot of stuff that we're doing in Shishto, we're really focusing on urine because it's so easy to collect. But for a home test, I can see, different circumstances that actually building with saliva is actually quite powerful. Just culturally, people put urine either in something that's ultra pure, which is not true, or they put it in a scenario where it's actually, it would be an issue. to even bring it inside a room and say run a test on a dinner table, or where else, drawing room, where would you, I mean, would you run that in the bathroom? Where would you actually run this test?
Because I was just comparing this with the pregnancy test. I mean, nobody finds that really difficult to go with that. It's because it's just a matter of, I think, accepting what is available.
Yeah.
But
No, I think that's
yeah,
a great
that's-
counterpoint.
Yeah.
And then it also makes for the scenario of just very directly, then the bathroom is where you're actually running this test.
Yeah.
Which
It's
is
about
a-
getting comfortable with the test and the need,
Yeah.
and I think
Yeah.
yeah, it's a great project. All the best.
Yeah, thanks.
Okay, so it's midnight here. I think, was there anybody else who was planning to present today? Tyler, you recorded some of these, right?
Yes, these are recorded.
Yeah. So I think we'll continue these discussions. One thing I'll do this week before next week, I'll be in Senegal. And since I won't have childcare duties, I will have more time rather than less time in the field. And so it would be valuable if people want to do just one-on-one kind of office hours. I'll just post sometimes. We'll try to then, I have to check whether the time exactly on Monday 10 p.m. would work. But I think what might be more useful is to Pranav to do a sit down with one of the sickle cell team. Let's do another one, Peter, Emma, if you guys want to sit down on kind of what might be the deliverables for the end of the class. And then similarly for Akhil itself, just, these could be just brainstorming sessions. So let's coordinate on Discord. And then both this week and next week should be possible, but it would be valuable to do those sooner rather than later. And especially, I think on the experiment side, I can see for the sickle cell front, Pranav, since you actually have access to these samples, I would love to run a quick training session for you to kind of, we can do a live blood cell imaging to really show the team how good dynamic time-lapse images you can do currently, even with a full scope, which I'm assuming all of you have. Does everybody here have a full scope, or is that not true? Okay, if that's not true, Tyler and Anton, I think, Tyler, I'm going to give you around 20-ish or so full scopes, and then we will use the class funds to actually ship full scopes to everybody wherever they are. Let's just make sure we do that. Yeah, I think it's, I find it ironic that you guys don't have a full scope, so let's fix that. Yeah.
Sounds great.
Yeah.
I'll send out a Google form via Discord so folks can put in an address and then we'll figure out how to
just
get
put
it
an
there.
address and we will ship it to you guys. I don't know how many days, months, or years it might take to reach you, but it will eventually reach you. And then I think, Tyler, the way we ship, we shouldn't put outside that it's a microscope. We should like send it as a document or something. If we say it a microscope, it's never going to make it into India.
That sounds great.
Yeah, I just looked there. All microscopes in India have like 40% custom duties, and in Brazil that have 70% custom duties.
Whoa.
So it's insane. Like for a $2 microscope, suddenly you have to try figuring out paying significant costs around. So I think in general, things will get stuck. So let's just send it as a document.
Sounds great.
Which, I mean, we're technically doing, we're not doing anything illegal. It is a document. So yeah. All right, bye everyone. I'll see you guys soon. This was so much
Good night,
fun.
everyone.
Thank you.
Awesome project.
Thanks. Good session today.
You
Take
guys
care.
are all amazing.